Next up is our second proceedings presentation by Mathias Cardner from the ETH in Zurich. And he will talk about inferring signaling dynamics by integrating interventional with observational data.
Thank you. All right. Hello, everyone. My name is Mathias, and I'm in Nico Berenwinkel's group. So today I'd like to talk to you about a project that we've done together with Nathalie Mayashala and Gerhard Cristofori at the University of Basel. And I'll begin with the biological motivation for this project, the experimental design that in a very direct way influenced how we chose to develop a model or extend a model. I'll introduce I will give you the nested effects model, a standard nested effects model for perturbation data, and then show you how we extended it in order to integrate time series non-interventional data into that framework. So the biological backdrop is epithelial to mesenchymal transition, EMT. So this is a process by which epithelial cells become migratory, mobile, and it's a part of development in general, but also has a role to play in cancer metastasis. And the aim was to infer the signaling network coordinating EMT, signaling network now of regulators such as transcription factors and microRNAs. So the experimental design is really, it's two experimental setups, and they are orthogonal in a way that I'll describe. So it's really two different setups. And the first one at the very top is a time course. It's an EMT time course where I should say it's murine mammary gland cells. These are mouse cells that are in epithelial state, and they're treated with transforming growth factor beta to undergo EMT. And they would reach a mesenchymal state after a certain time, and we have samples at time points during that transition. And these are samples that are biologically duplicated and measured using RNA sequencing. So that's the first setup, and the second experimental setup is a perturbation experiment, and actually 59 perturbation experiments targeting 59 different regulators. So those are transcription factors and microRNAs, which are separately perturbed, and we know that they're crucial for EMT because if you perturb them and treat the cells with TGF-beta, the cells don't reach the mesenchymal state. So they're stuck at some kind of intermediate phenotype, which is not mesenchymal. And in contrast to the above design here, we don't have any time resolution in the sample. So we have no time series. We only have a snapshot at 4 days. And that's, again, biologically duplicated, measured using RNA sequencing. So that's what I mean when I say orthogonal experimental design, is that we have time series with no perturbation, and we have perturbation with no time series. So the model framework that we chose to use for this project was a nested effects model. And this is, I'll just give you this sort of overview. This was published by Markovets et al. In 2005. And the idea of a nested effects model is that the signaling nodes, sort of the regulators, in our case transcription factors and microRNAs, are proteins and microRNAs, right? So they are non-transcriptional. If you measure the gene expression, you would not get information directly about protein abundance, and you wouldn't get any information about post-translational modifications, which are crucial in signaling in mammals. And that's why we model them as hidden nodes. So these are the signaling nodes, or S nodes, that are hidden. They're not directly measured, but what we do measure is the gene expression profile. So this is RNA sequencing data giving us the effect genes, so the E genes, I will call them. And the assumption driving this network inference is really that we assume that each effect gene is regulated directly by at most one of these signaling nodes. So that doesn't mean that it cannot respond to more than one perturbation, but if it does so, then the assumption is that that effect is mediated through another signaling node. And that's how we drive the network hierarchy reconstruction. So to just give a toy example of this, let's say we have 4 signaling nodes that we perturb individually, and at each point we measure the transcriptome and look for differential expression here in purple. So we have some stimulus on this pathway. We have TGF-beta in our case, and we've perturbed this signaling nodes, and we see a differential expression in these 3 genes. We would draw the conclusion that this is the direct regulator of those genes among the signaling nodes we have. And we continue perturb the next signaling node, and we still see those 3 effects, but we now see 2 more. And since this is a superset of the original set, we now believe that this is upstream of that signaling node in the network. And we continue like this. We perturb another one. We see new effects at another part of the transcriptome. It's a sort of a different branch, if you will. And then at the very root, we would find differential expression across. So I should say this is not the entire transcriptome. We choose a part of it that actually responds to at least one perturbation and so on, filter for relevance. And we fit this now to our data. So this is so far only the perturbation data, the standard nested effects model, which we've fit. And we've done resampling using Bootstrap and Jackknife to get a confidence in the edges. And I'm showing you here the consensus network, where transcription factors are in blue and microRNAs are orange. And from a biological point of view now, I should say this is a directed acyclic graph, and it's hierarchically, topologically ordered, so all the edges are going downwards. And all the transitively closed implications are removed. So I'm showing you a cleaned up version. And biologically speaking, we were a bit surprised to see some of these early EMT factors, SMAD4, ZAB1, and 2, to be fairly far down this hierarchy because they're sort of believed to be early. And it should be said that all these factors are EMT factors. They're sitting at the very top of the EMT program. Nevertheless, we wanted to look into the prediction of the fit of the nested effect model. So we performed validation experiments. So Natalie did these luciferase reporter assays of certain transcription factors, where she actually measures the activity of the transcription factor under various perturbations. So these are, each panel here is a luciferase reporter assay. Let's look at the one for SMAD4, for instance. I'm showing you the volcano plot under perturbation of various regulators. And of course, you would see SMAD4 here being very significantly affected by its own perturbation and also negative log fold change. The coloring now comes from the nested effects model, whether it would predict an effect in green or whether it would predict a non-effect in orange. And ideally, you would want to see all the green above this FDR cutoff and all the orange below. So it's not as clear cut. Remember, this is a difficult sort of hidden causal network inference. But nevertheless, the precision and recall is 75% and 63% respectively. So now I'd like to move on to the model extension that we performed to bring in this time series data into the network. And I'm showing you here the principal component analysis of the time series samples, so the time course experiment. First principal component captures 70% of the variance, and it almost perfectly chronologically orders these samples in increasing order. The clear exception here is 60-hour time point, which seems to revert in a way, and I'll get back to that in a few slides. And the intuition now is that we want to take these time series samples and really map them onto the static network that we have inferred using the nested effects model. And in that way, we want to assess how the function, how the activity is progressing through this network. How is the activation looking during the time series? And now we really have the reverse problem from before. So now we don't have any perturbations anymore, but we do have the inferred static network with all these attachment of E genes. And what we're looking at is the differential expression between 0 hours now to start with against 96 hours. And 96 hours is always the baseline. That's the time point at which the perturbation experiments were sampled. And to start off, we would see a complete differential expression here, and you would assume that, or draw the conclusion that there is, let's say, there's differential signaling activity at this time point compared to 96 hours. And then you move on and you begin to see some of this differential expression clearing up. In specific, it would be the one at the very top of the network, which we no longer then judge as differentially signaling compared to the end time point, and we would therefore infer it as being active or being in a similar state. And we progress through the time series now at 48 hours, we see more clearing up, and we use these E genes as a proxy for what's going on with the signaling node until we reach a state where we're in a completely activated signaling network. So that was the, let's say, graphical intuition. I'll just 2 slides to show you a bit more of the details of how we do this. We have now a time point, small t, which is before the steady state time point. And we're given now the signaling network, the static network inferred by the nested effects model, the attachment of E genes to that network, the differential expression profile of p-values from the time point t versus the steady state. And from that profile, we can fit and estimate the density function of the alternative hypothesis of p-values, which we use later on. And now the purpose is to be able to compute the log likelihood of an activity pattern that I'm going to call K. So K is really just a binary vector telling you which node is active, which one is not. And this turns out to be a sum over all the E genes, so the effect reporter genes, and you're summing up the log density of the p-value for that gene, if and only if the model and so the activity pattern would predict an effect or not at that given gene. And we don't need to search the full space of these binary K vectors. We can constrain it to solutions that would conform with the network that we have. And by conform and be consistent with, I mean that they respect the transitively closed property of that network. And that allows us to actually reformulate this in a linear, integer linear program, and we then can solve that using a branch and bound algorithm. In our case, we used LP-solve for this. So we fit this to our time series data, and now I'm showing you for graphical representation the consensus network in every case here. But again, this is done over the bootstrap and jackknife resamples. I'm just using this for presentation. And the green color now will, like in the toy example, it will indicate activity of a signaling node. So as we begin the time series at 24 hours, 36, 48, we begin to see an activity, sort of an activation at the top of the network, which progresses downwards. And then at 60 hours, we have what looks like a reversal of this progression. And that's sort of in line with what the principal component analysis suggested for this sample. It's also in line with what other groups, other labs have seen in proteomics experiments in EMT systems. There's appeared to be something happening at 60 hours, which could be, we hypothesize it's an obstacle that the cells need to overcome in order to progress. It could also be that the PCA and this directed acyclic graph is not able to capture something that's nonlinear and has feedback loops and so on. And from a biological point of view, what we get from this, you remember I talked about these early EMT factors at the start, and when we look at this at 48 hours, we see that those nodes are just beginning to get some calls for activity. It's maybe difficult, it's a very small figure, but you see that there is some evidence for activity calls in those nodes at 48 hours. So the conclusions are that we developed or extended this model to integrate non-interventional time series data into a nested effects model framework, which is learned from perturbation data. And the key point here and the real motivation for doing this is that the fully factorial design, if you will, where you have time series data of perturbation experiments really blows up in terms of resource cost, right? Whereas the orthogonal design, where you have just the time series on its own and the perturbation data without time series, is much more common. I'm sure you all know this as well. And this is a way to take those two types of data and bring them together in the framework of a nested effects model to do this kind of inference. And this is implemented as an R package on GitHub. And I'd like to acknowledge collaborators Maren Diepenbroek, Mira Saxena, and Robert Ivanek. And I want to thank my professor, Nico Berenwinkel, and his group, in particular, Jack Kalpus, David Seifert, and Martin Pirkel. And thank you very much for your attention.
Thank you very much. Are there questions from the audience? Yes, please.
Hello, thanks for the talk. So in the result that you mentioned about SMED showing up at the bottom of the EMT network, do you think that the NEM could account for feedback mechanisms that maybe SMED has some regulators, but also regulates some other regulators? Could you elaborate on that, please?
Right. So, I mean, in principle, it cannot account for feedback loops. And the reason is that you're looking at the transcriptomic readout here, as you're really looking at what did the perturbation do to the transcriptome? Like, so what are the global effects on the transcriptome when you remove this signaling node? And if you don't have an actual time resolution in your perturbation data, you're just faced with that at the steady state time point. This is what happened. And therefore, you cannot really resolve any kind of feedback going on in the system unless you do have time there. And there are some dynamical nested effects models which are designed for that case, where you do have time in the perturbations data. So that's one potential place to resolve it, I think. Yeah.
Yes, please.
There's so I have a question about these, how you built the nested effects model. So if you take as an example that a perturbation will, let's say, cause an effect at 10 of your readouts, and then you have another perturbation in which 9 of these readouts, let's say, actually respond, and let's say 5 others, then I would intuitively assume that this is probably upstream, and the 10th node simply was maybe a measurement error or something like this. So how? But this sort of suggested a little bit like you rely on the fact that you have actually unions of these sets.
That's a great question. Thank you for bringing. I should have mentioned this. So this is a toy example, but the full model is, of course, a probabilistic model where you have error models and accounting for this kind of thing. So it's not like perfect. It's not perfect subsets or anything like that. But this, you're looking for the network which is the most likely network with respect to that assumption. Yeah. Thanks.
Any more questions from the audience? Yes, please.
Great talk. EMT is a reversible process. So can you comment on if you were to incorporate the MET with the EMT data, how will you do this?
Right. Yeah, that's a very good point. We haven't got that kind of data, but it would be very interesting to have it, right? Or yeah, you would then have to reverse it using some stimulus and do sort of the same model. But just from a data point of view, theoretically, I'm not quite sure how to reverse it in this way. But it's a good point. It's a reversible process. That's why it's important for metastasis formation as well, because the cells would undergo EMT and become mobile, migrate, and then undergo MET, right, and seed a daughter metastatic outgrowth. That's a good point. Thanks.
Yes, please.
Yeah, very nice talk. Thank you. Yeah.
Thank you.
I was wondering, if the nested effect model could be applied also to like nodes that you don't know about. So if you don't know a gene, or if you could discover some, common mode with it or not.
Well, so there are two things come to mind. The first one is an extension in 2009 by Vasque et al. It's called a factor graph nested effects model, where they do exactly that. They propose regulators that were not perturbed, and they say this is sort of candidate list of other players in this network that you sort of could validate or look up, because it's obvious that you need to restrict this quite heavily when you're going to do the perturbation experiments. In our case, we had 59 of these EMT factors, but there are very many more, right? So we're taking the ones from a screening of 1,500 potential regulators, and we're taking those that blocked EMT in our case. But it's a constrained subset, yeah.
Any more questions from the audience? Otherwise, I would like to thank you again for the nice